For many years, medical professionals and scientists have observed a perplexing and troubling connection between two serious neurological conditions, Alzheimer's disease and epilepsy. Clinical evidence has consistently suggested that individuals who live with epilepsy face a notably higher risk of developing Alzheimer's disease as they grow older. Conversely, it is also well documented that individuals diagnosed with Alzheimer's disease frequently experience seizures, which are the hallmark of epilepsy. This overlap has prompted extensive research, as understanding the underlying mechanisms could unlock new pathways for treatment and prevention for millions of people worldwide. These conditions not only profoundly impact the individuals affected, but also place a significant emotional and economic burden on families and healthcare systems globally. Finding common ground is therefore a critical priority. Recent groundbreaking research, particularly from a dedicated team of scientists at the University of São Paulo, USP in Brazil, is now shedding crucial new light on this intricate relationship. Their insightful study, supported by the São Paulo Research Foundation, FAPESAP, and published in the Esteemed on February 22, 2025, has provided compelling evidence from animal models. This work strongly indicates that there may indeed be a shared biological factor contributing to both Alzheimer's disease and epilepsy. This Brazilian research is a significant step forward, potentially reshaping our fundamental understanding of how these two distinct yet interconnected neurological disorders develop and progress within the human brain. The findings are already attracting considerable attention within the scientific community for their innovative approach. At the heart of this new understanding is a concept known as brain insulin resistance. The USP researchers have demonstrated that altered insulin signaling within the brain appears to be a critical factor affecting both conditions. It is important to clarify that this is not necessarily the same as the systemic insulin resistance commonly associated with type 2 diabetes, which affects the body's overall ability to process sugar. Instead, this refers to a specific problem where the brain cells themselves become less responsive to the hormone insulin. This impairment in the brain's ability to utilize insulin effectively can disrupt normal neuronal function and energy metabolism, leading to a cascade of detrimental effects. This focus on the brain's unique insulin mechanisms is a pivotal shift. Understanding this profound link is of paramount importance, especially in the context of our aging global population. Alzheimer's disease, a progressive neurodegenerative disorder, currently has no cure and remains one of the most significant public health challenges of our time. Similarly, epilepsy, characterized by recurrent seizures, can be debilitating and affects people of all ages. The discovery that brain insulin resistance could be a common denominator offers a glimmer of hope. It suggests that therapeutic strategies aimed at addressing this specific type of insulin dysfunction might hold the key to developing more effective treatments, potentially benefiting patients suffering from either or both of these devastating conditions. The implications for future public health strategies are truly substantial. Insulin, a hormone often associated with blood sugar regulation, plays a far more extensive and critical role in the brain than previously understood. Within the intricate neural networks of our brains, insulin is vital for numerous functions essential for cognitive health. It actively supports neuronal survival, helping to protect brain cells from damage and degeneration. Furthermore, insulin is deeply involved in synaptic plasticity, the remarkable ability of synapses, the connections between neurons, to strengthen or weaken over time. This process is absolutely fundamental for learning new information and forming memories. When the brain begins to develop resistance to insulin, its cells no longer respond efficiently to insulin signals. This impairment means that these crucial supportive and regulatory processes can falter, leading to significant disruptions in overall brain function. The concept of type 3 diabetes has emerged in scientific discourse to describe a state of severe insulin resistance specifically occurring within the brain. This term underscores the idea that the brain can experience its own form of diabetes-like condition, distinct from the more commonly known type 1 or type 2 diabetes that affect the body's glucose metabolism. A crucial aspect of this is that central insulin resistance can arise even in individuals who do not exhibit symptoms of systemic diabetes. This means a person might have perfectly normal blood sugar levels throughout their body, yet their brain could be silently struggling with impaired insulin signaling. This distinction highlights the complexity of brain metabolism and points to the need for diagnostic tools and treatments that specifically target brain insulin function. The consequences of this impaired insulin signaling in the brain are serious and far-reaching, as highlighted by the research. When brain cells become resistant to insulin, it can lead directly to neuronal damage, compromising the health and integrity of the very building blocks of our nervous system. This resistance also contributes to defective synaptic plasticity, particularly in regions like the hippocampus, which is the brain's primary center for memory formation and consolidation. As a result, learning abilities and memory recall can become significantly impaired. Furthermore, the brain's cholinergic system, which relies on the neurotransmitter acetylcholine for functions like attention and memory, can also be negatively affected. Chronic neuroinflammation, a persistent state of inflammation within the brain, is another potential outcome, further exacerbating neuronal dysfunction. These detrimental effects of brain insulin resistance are strikingly similar to, and indeed can directly contribute to, the pathological hallmarks universally recognized in Alzheimer's disease. For example, research suggests that an environment of brain insulin resistance can promote the increased production and subsequent accumulation of beta amyloid proteins. These proteins aggregate to form the sticky plaques that are a defining characteristic of Alzheimer's brains, disrupting communication between neurons. Additionally, impaired insulin signaling is linked to the hyperphosphorylation of tau proteins. These abnormal tau proteins then form neurofibrillary tangles inside neurons, another key pathological feature of Alzheimer's, leading to cell death. This shows a clear pathway from insulin problems to the devastating changes seen in the disease. The dedicated researchers at the University of São Paulo employed highly innovative and meticulous methodologies to investigate the intricate connections between brain insulin resistance, Alzheimer's disease, and epilepsy. A significant part of their study involved the use of animal models, which allow scientists to study complex biological processes in a controlled environment. To create a model of Alzheimer's disease pathology driven by insulin issues, the team microinjected a substance called streptozotocin directly into the brains of rats. Streptozotocin is a chemical known for its ability to induce insulin resistance, and by administering it intracerebrally, the researchers could specifically target and disrupt insulin signaling pathways within the brain tissue. This careful approach was crucial for isolating and observing the direct consequences of brain insulin resistance on neurological health. One of the most compelling and significant outcomes of this experimental setup was observed in the behavior of these rats. The animals that had brain insulin resistance induced via streptozotocin, thereby modeling key aspects of Alzheimer's disease, also began to exhibit seizure-like symptoms. Specifically, these rats showed an increased susceptibility to seizures when they were exposed to high-intensity sound stimuli, a common method for testing seizure thresholds in animal models. This was a crucial piece of direct evidence from their experiments. It strongly suggested that the state of brain insulin resistance, induced to mimic Alzheimer's pathology, could concurrently render the brain more vulnerable to epileptic activity. This finding powerfully reinforces the hypothesis that insulin dysfunction is a common thread connecting these two disorders. In a complementary line of investigation, the USP team also turned their attention to a specialized strain of rats known as Wistar audiogenic rats, often abbreviated as WARS. This particular strain has been genetically developed over many years, including at FMRP-USP, specifically for epilepsy studies because these rats are inherently susceptible to seizures triggered by sound. The researchers examined these epileptic rats for molecular markers typically associated with Alzheimer's disease. Remarkably, they found that the brains of these WARS, genetically prone to epilepsy, did indeed show Alzheimer's-like changes. These included the hyperphosphorylation of tau protein, a key element in the formation of neurofibrillary tangles, and a noticeable decrease in the number of insulin receptors in the hippocampus, a brain region critical for memory and often severely affected in Alzheimer's disease. further underscoring the critical role of intact brain insulin signaling. Sulin Santos Alves, a PhD student and the first author of the published study, highlighted another striking observation. The researchers administered a single dose of a drug designed to acutely induce brain insulin resistance to rodents that were already exhibiting characteristics of both epilepsy and Alzheimer's-like pathology. The results were clear and concerning. This intervention led to a worsening of their memory functions and, simultaneously, an increase in both the frequency and severity of their seizures. This experimental finding strongly reinforces the idea that disrupting insulin pathways in the brain can exacerbate the symptoms of both conditions. Alves also noted that the genetic background of the animals can influence how they respond to such interventions and the overall progression of the diseases. The comprehensive findings emerging from the University of São Paulo study represent a truly significant breakthrough in our understanding of complex neurological disorders. This research is particularly noteworthy because it is among the very first to clearly demonstrate a direct and tangible link between the state of brain insulin resistance and an increased susceptibility to seizures. By identifying this specific biological mechanism, the study provides a compelling explanation for the long-observed clinical overlap between epilepsy and Alzheimer's disease. Gaining insight into such a fundamental connection is a monumental step forward. It not only validates previous hypotheses, but also opens up entirely new avenues for focused research, and importantly, for the development of innovative therapeutic interventions that could target this shared pathology. Professor Norberto Garcia Carrasco, the corresponding author of the study and a respected professor at FMRP-USP, has emphasized the broader implications of these findings for how we approach Alzheimer's treatment. Alzheimer's disease is notoriously complex and multifactorial, with numerous contributing factors. Current pharmacological treatments, which primarily include anticholinesterase inhibitors and anti-glutamatergic agents, unfortunately offer limited efficacy and mainly address the symptoms rather than the underlying causes of the disease. The discovery that brain insulin resistance is a key player strongly suggests that a more comprehensive therapeutic strategy is urgently needed. We must move beyond the paradigm of single-target drugs and consider approaches that address the multifaceted nature of the disease, including metabolic dysfunctions within the brain. Looking ahead, the University of São Paulo Research Group is poised to continue their pioneering work and delve even deeper into this critical area.